In this video, I will walk you through the steps of genomic DNA extraction from cancer cells using the QIAamp kit. The genomic DNA will then be used later as template in PCR reactions. When working with eukaryotic cells, such as cancer cells, our primary focus is to keep them uncontaminated. Therefore, we use gloves to prevent any skin cells to fall into our dishes and use ethanol to sanitize the surface of our hands as well as the laminar flow hood. The hood is a specific device that we use to keep any aerosol that could contain cells from contaminating the dish. The cells were currently in 37 degrees Celsius culture at 5% CO2. And let's have a look at the cells and their density. As you can see here very nicely, we have about 90 to 100% cell density. The cells grew in a monolayer and covered a whole dish so that we have the maximum amount of cells for genomic DNA extraction. So now we're transferring the cells into our laminar flow hood. And again, we want to make sure that we don't introduce anything under the hood that could contaminate the cells. So the first step is to remove any media that the cells are currently growing in. You see it's slightly orange rather than red, which indicates a more acidic pH, which indicates a high cell number. What you see me now doing is to sterilize the surface of my PBS or phosphate buffered saline solution. This is a sterile salt solution that we use to wash the cells. So the first step that we do here is to take 10 milliliters of PBS and add it to the cell culture dish to wash off any dead cells that are on top of the monolayer. Since the cell culture medium contains, besides vitamins, salts, and glucose, also fetal bovine serum, which could potentially introduce foreign DNA, we want to do this twice to really make sure that we have an absolutely clean solution of the cells. Once we've done that, we adjust our pipetter to add 200 microliter of Buffer AL, which is the so-called lysis buffer, to our plate. To remove the cells from the surface, we're using a cell scraper, which is packaged sterile. Other methods to remove the cells from the surface include the addition of an enzyme that breaks up the bonds between the plate and the cells. But since we're not concerned about breaking the cells open with our cell scraper, since they're anyway in a lysis buffer, we can go ahead and use the more forceful solution. At this point, the lysate has more of a goo-like or gel-like consistency that we're now transferring into a pre-labeled tube. So initially we transferred 200 microliters, and then after we transfer these 200 microliters, we will go in 20 microliter increments to really measure how much volume we're transferring, because there was most likely a little bit of residual PBS in the dish that we're now transferring. At this point, it's very important that we count how many times we transferred 20 microliter so that we can calculate how much additional PBS we have to calculate. So 1, 2, 3, 4, 5, 6. Maybe we stick with counting 5 since this is not completely 20 microliters here and mostly bubbles. So we in total had 5 times 20 microliters that we transferred, which counts to 100 microliters. So we would adjust our pipette to 100 microliters PBS to get it up to a total volume of 400 microliters. Lastly, we want to adjust the pipette one more time to 20 microliters in order to add the Proteinase K, which takes care of all protein residues that can potentially even interfere with our DNA and degrade it, which we really don't want. So now that we have our lysate with Proteinase K ready, we want to mix it very well by vortexing it for about 15 seconds and then add the mixture to a heat block that is at 56 degrees because this is the temperature that the Proteinase K will work. After 10 minutes incubation, we're ready to so-called precipitate the DNA, which means we add 200 microliters of ethanol to the solution. That ensures that the DNA becomes in a state that can stick to our final column. Once we've done that, again, we want to mix very well to really make sure that the ethanol gets to the whole lysate. And then we are ready to transfer the lysate over to our column. For that, we adjusted a pipette to around 800 microliters and take about 1 to 2 times transfers into the column by not touching the outside or the rim, but really adding it to the membrane itself. Then we're ready to ensure the flow through of our lysate over the column by centrifugation. Since we only have one sample, we want to ensure that we have a counterweight in our centrifuge to keep it perfectly balanced and not damage the centrifuge itself. So the centrifugation will be at 6000 times G or 800 RPM for 1 minute. While we're waiting for the centrifuge to be done, we can grab an additional clean 2 milliliter collection tube that is provided by the manufacturer and put it to the side so that we can transfer the column itself to our new clean tube. The flow through at this point can be discarded, and we only keep the column that can be kept at 4 degrees in the fridge over several weeks. And in our case, I'm gonna keep that for you guys ready to be used in the lab. Now that we're ready and excited to extract our genomic DNA and prepare the PCR reaction, I want to emphasize that the PCR is so sensitive and will amplify and use any genomic DNA as a template. So therefore, we're working throughout the lab completely with gloves, wash our hands before we get started to really prevent any contamination in our reaction of any genomic DNA that comes from us, our skin cells, or anything else. As you're getting to your workbench, you will find a bucket with ice that includes the column that I previously prepared. As a first step, when you're getting to the lab, you can grab the column out of the ice bucket and acclimate it to room temperature. The first thing that we will do is grab the AW1 buffer, which contains ethanol and functions as an additional wash buffer. We adjust the pipette to 500 microliters and add the solution carefully to the column's membrane without touching the membrane itself. At this point, I want to mention that we will have only one centrifuge in the lab, so everybody should be aware of what they're doing and do that quickly so that everybody can grab their column and add it to the centrifuge in a coordinated way. For this first centrifugation, we will have the centrifuge adjusted to 6000 times G and will spin for 1 minute. Again, it is very important that when we have an uneven number of tubes, we will have a counterweight to not damage the centrifuge itself. At this point, you see me scratch my head while I was waiting for the samples, which introduced contamination or skin cells to my gloves. So I went ahead and grabbed some ethanol to re-sanitize my hands and really prevent any introduction of foreign DNA. Alternatively, you can also change your gloves, which is fine too. So now that we're ready with our first centrifugation, you will see that all the wash buffer has flown through all the way to the bottom of the tube. And we're ready to add our second wash buffer called AW2. So if you haven't played with your pipette, you can just take it and use it right adjusted to 500 microliters. But before we add our buffer, we want to remove the column carefully and take our 500 microliter pipette to remove now the flow through and discard that. So we just suction it in with the pipette and throw the pipette with the liquid right into the trash. Now we can grab our Buffer AW2 together with our pipette adjusted to 500 microliters and add it carefully to the membrane of our column, again without touching the membrane or the outside of the tube itself. Now heading to our centrifuge, we load our sample and want to make sure that we adjust the speed to the maximum, which should be around 14,000 RPM or 20,000 G. And the time that we spin will be 3 minutes. At this point, we have some time to get our final collection tube ready. So we grab one of these tubes that are all sterile, drop them onto the surface to really only remove one, and carefully close it without touching the inside of the tab. We want to now write our group name or group letter, as well as the genomic DNA that we used, and the word gDNA on top of the tube as well as on the sides, together with the date, so that we have it perfectly labeled. You can also go ahead and locate the ultra pure water, which is RNase and DNase free and will be used for elution. Now we're going to the centrifuge and grab a final tube that is empty and will be used to collect any residual wash buffer that is left on the column in a final step of centrifugation. Therefore, we're taking out our tube and just switch the column from the full tube into the empty, fresh, clean collection tube and place it right back into the centrifuge. Again, we're spinning for 1 to 2 minutes, and after the centrifuge is stopped, you will see that there is indeed a small amount of wash buffer in the bottom that contains ethanol, which would prevent our PCR to work if we weren't removing this. So we can discard now both of the collection tubes and transfer the column to our final collection tube, which is a 1.5 milliliter tube that we previously labeled. We now want to adjust our pipette to 200 microliters and transfer this amount of volume of the ultra pure water carefully onto the column. We then want to make sure that our column is incubating at room temperature for at least 5 minutes before we start the centrifuge. Once 5 minutes are over, we head over to the centrifuge, adjust now the speed to 6,000 times G again, and we'll centrifuge for 1 minute to elute the DNA. At this point, it is good practice to take out the tube and have a look at the collection tube before discarding the column to make sure that there's indeed 200 microliters at the bottom of the tube. If the volume is smaller than that, you most likely have pipetted the wrong amount, and you can just go ahead and add the additional amount to the column and respin it. If it is too much, you most likely have not removed all ethanol, and we will see if downstream assays will be inhibited by that. Now we're ready to put the genomic DNA on ice and measure the concentration for our downstream assay.